Scientists measured the chemistry locked inside a giant ground sloth Charles Darwin himself helped make famous. And the reading did not match the peaceful leaf eater every textbook described. The animal is Mylodon darwinii, a giant ground sloth from the Ice Age, and one of the most famous extinct creatures we've got, partly because its bones and even its skin turned up in a cave in Patagonia. Darwin was there for the early discovery. His name is literally stitched into the species. And for nearly 2 centuries, one thing about it was considered settled: what it ate. Hold on to that word, herbivore, because in a moment, the chemistry stops agreeing with it. So, picture the animal first. Mylodon was bulky and ground-hugging, roughly the size of a small car, nearly 3 meters long. It had heavy clawed forelimbs and a broad, blunt muzzle, jaws built for one job: cropping vegetation. This was not a runner, not a hunter. This was a slow, low-slung mower of the cold plains. And that's exactly the picture science painted: a gentle giant grazing across the chilly grasslands of South America, stripping leaves, chewing through the day, doing that for thousands of years until the species vanished roughly 10,000 years ago. A peaceful ending to a peaceful life. Why did everyone agree? Because the body backed it up. The teeth were the flat, grinding kind you'd expect in a leaf eater. The gut was huge the way it needs to be to ferment tough plant matter. Every line of anatomy pointed the same direction, and every textbook wrote it down the same way. The bones said herbivore. Full stop. Picture this animal for a second. A clawed, car-sized beast, built for stripping leaves. Now ask yourself what could possibly break that picture. Here's the first crack. Most extinct animals leave us bone and nothing else. Bone fossilizes, soft tissue rots away. But Mylodon is strange. In one place, more than bones survived. Deep inside Cueva del Mylodon, in Chilean Patagonia, researchers pulled out preserved dung, preserved hair, even patches of skin, dried and kept in the cool, still sediment of the cave floor. Thousands of years old, and still there. Stop and sit with how rare that is. We're not talking about a mineral copy of an animal. We're talking about actual sloth tissue, the real stuff, hair you could hold. And that changes what's possible, because tissue isn't just a body, it's a record. When an animal is alive, everything it eats gets written into its flesh at the chemical level. Bone can tell you the shape of a creature, tissue can tell you what it was actually doing, day to day, meal to meal. And with Mylodon, for once, the tissue was still readable. The preserved tissue meant researchers could measure nitrogen isotopes, the chemical fingerprint that tells you exactly where an animal sat on the food chain. But bulk readings are noisy. The environment and the plants themselves smear the signal. So, paleontologist Julia Tejada's team went finer. And what they chose to measure is where this whole case turns. Nitrogen isotopes. That was the fingerprint. In any animal's body, nitrogen ratios climb as you move up the food chain. Plant eaters read low, meat eaters read high. Eat the grass and you sit near the bottom. Eat the thing that ate the grass and your number jumps. So, in theory, you take a scrap of preserved tissue, measure the nitrogen, and you know where the animal fed. Simple, except it isn't. Here's the problem. The baseline moves. Soil chemistry shifts it. A dry climate shifts it. Drought, the type of plant, even the fungus in the ground. All of it nudges the nitrogen values up or down before the animal has eaten a single bite. So, a bulk reading, the raw number off a whole sample, can lie to you. It can make a strict herbivore look like it dabbled in meat, just because the plants it ate were already running high. That noise is exactly why Julia Tejada's team refused to stop there. Here is the move that makes this whole story trustworthy. And it is simpler than it sounds. Instead of measuring the nitrogen in the sample as one lump, they measured it in individual amino acids. The building blocks of protein, read one at a time. Why does that matter? Because some amino acids track the diet itself. Others track the environment, the baseline. Measure them separately, and you can subtract the noise. The soil signal goes in one column, the food signal goes in another. What's left is a clean read of what the animal actually ate, stripped of where it lived. Julia Tejada, the paleontologist who led the analysis, pushed for exactly this finer method, not the blunt tool, the precise one. And this is where a good scientist gets nervous, because a fancy new method can invent patterns that aren't there. If the technique were flawed, every sloth you tested would come back looking strange. So, Watch what the control animal does instead. The team ran the same amino acid test on a related sloth species. Same lab, same procedure, same instruments humming under the fume hood, and that relative came back cleanly herbivorous, low nitrogen, right where a leaf eater should sit. Textbook. That single clean result does something important. It proves the method works. It shows the tool isn't just spitting out weird numbers for everything ancient and degraded. When you feed it a plant eater, it tells you plant eater. So whatever it says about our giant sloth, it isn't an artifact of a broken technique. The control behaved, the method held, which isolates the anomaly to one animal, Mylodon. And here the gentle giant label starts to feel loose, because the amino acid signature in Mylodon did not read as a pure herbivore. This is a creature built like a leaf stripper, the broad muzzle, the cropping jaws, the gut the size of a barrel, carrying a chemical signal that a leaf-only diet simply cannot produce. The bones said herbivore. The molecules said something else. The nitrogen values in Mylodon did not point to a plant eater. They pointed to an omnivore, an animal that at least sometimes ate animal protein. A car-sized beast with a leaf stripper's jaws, carrying a chemical trace no salad can explain. And the first question everyone asks next is the wrong one. Did Mylodon hunt? Did Mylodon hunt? That is the wrong question. And answering it honestly is where this story earns its weight. The nitrogen signature pointed to an omnivore, an animal eating animal protein, at least some of the time. But that is not a predator reveal, and the scientists are careful to say so. Here is what the chemistry actually shows. Mylodon's diet was more flexible than the herbivore label ever allowed. Not a leaf-only mower of the plains. An animal that, at least sometimes, ate animal protein, likely opportunistic feeding on animal protein when it could get it, scavenging a carcass, cleaning up what a bigger predator left behind, eating whatever the cold world put in front of it. That is the thing that doesn't match what they thought. A car-sized beast with cropping jaws, carrying a chemical fingerprint no salad can explain. But let's be exact about the line here. This is not a claim that Mylodon hunted. The bones still show an animal built for stripping vegetation. The broad muzzle, the flat grinding teeth, the heavy gut. Nothing about the skeleton says killer. The clawed forelimbs that look so fearsome were tools for pulling down branches, not tearing flesh. The molecules widen the menu. They do not turn a sloth into a lion. And now the caveats, because this is the part most stories skip. So pay attention to what the evidence does not prove. Measuring nitrogen in individual amino acids is a powerful method, but it's young. Applying it to ancient tissue that sat in a Patagonian cave for thousands of years, raises real questions. Did preservation shift the chemistry over time? And there's a bigger one. This reading comes largely from individual animals. One sloth is not a whole species. Maybe this Mylodon was unusual. Maybe the diet varied by region, by season, by desperation. The signal is real. What sits behind it is still being argued. So, hold on to both halves. The finding is published. It is peer-reviewed. It is not a rumor or a headline. It's science that cleared the bar. But peer-reviewed doesn't mean settled. The exact behavior behind the chemistry remains open, actively debated by the people who study these animals for a living. That tension is not a weakness of the work. It is the work. And this is where one sloth stops being about one sloth. If a supposedly gentle giant was bending its diet to survive, the whole picture shifts. Picture Ice Age Patagonia, cooling, drying, food thinning out. Megafauna crowded onto shrinking ground, competing for every mouthful. In that world, being a strict herbivore is a gamble. Being flexible is an edge. The nitrogen climbing the food chain in Mylodon hints that these giants were not passive. They were improvising, which complicates the tidy story of why they vanished. For a long time, the extinction of the Ice Age giants got explained in clean lines. Climate, humans, take your pick. But if these animals were already adapting their diets to hang on, survival was messier than the textbooks let on. More opportunistic, more like a struggle you can almost feel. The bones said herbivore. The molecules said something more complicated. And that gap, the space between what a skeleton looks like and what a body actually did, is exactly where the past keeps surprising us. Think about what this means for every gentle giant we thought we understood. The bones said herbivore. The molecules said something else. And somewhere in a museum drawer right now sits the next creature whose chemistry refuses to match its skeleton.